Hello, this topic is name is components of cell. This is for NDA biology. Let's start. Here I have put the image of different cell organelles. It is same as I have put in my last slide. Nucleus, nucleolus, you can see this, endoplasmic reticulum, smooth ear, rough, this is rough ear, this is smooth ear, Golgi apparatus, mitochondria, centriole, cytoskeleton, cell membrane. Okay. Now this is the division of cells. One cell, where one cell consists of different organelles. First cell, which is divisible into cell envelope and protoplasm. It is the outer covering. Cell envelope is of two types. Animals, plant cell and bacterial cell, even fungal cell have cell wall. Okay, while in animal cell, the outer covering is cell membrane. So cell envelope is of two types: cell wall, cell membrane. Protoplasm consists of cytoplasm and nucleus. Okay. Cytoplasm is again divisible into two parts. One is cytosol, which is exclusively the solution part, and the cell organelles. Cytosol consists of ground matrix, ions, proteins. Cell organelles are Golgi apparatus, lysosomes, ribosomes, peroxisome, mitochondrion, cytoskeleton, centriole, vacuoles, ER or endoplasmic reticulum. Nucleus consists of nuclear membrane, which is double membrane having pore in them. Nucleoplasm, chromatin or DNA, RNA and proteins are present. The proteins are especially the histone proteins. Okay. These are the image of some separate cell organelles. Here you can see chloroplast, Golgi apparatus, nucleus, mitochondria, lysosome, ER. Okay. First come to nucleus. Nucleus is the control center consisting of DNA. Okay. Nuclear membrane is double membrane structure. Question often comes, name some cell organelles having double membrane. One of them is nucleus, another is mitochondria, and third one is chloroplast. Okay. Separating the nucleus from cytoplasm, composed of inner and outer membrane. Pores within the envelope are present, which facilitate the transport of RNA and proteins between the nucleus and cytoplasm. Nucleolus is non-membranous bound region. Nucleolus is a region without any membrane within the nucleus, responsible for producing ribosomes. rRNA, especially ribosomal RNA, are generating inside the nucleolus. You can see this is the image of cell nucleus. It is double membrane. This is outer membrane, and this one is inner membrane. Inner membrane. Okay, and this central part is called nucleolus. It is a dense region without any membrane, and you can see thread-like structures are there, DNA, and each membrane are separated by nuclear pore. Okay, this is nuclear envelope, and whole structure is cell nucleus. Now come to second organelle, that is ribosome. Ribosomes are present both in prokaryote and eukaryotes. Composition. Ribosomes are made up of ribosomal RNA, that is rRNA, and proteins. When rRNA and proteins are both present, it forms an enzyme that is called ribonucleoprotein particles. Okay. Subunit, it consists of two distinct subunits. One is large subunit, one is small subunits. Small subunit is responsible for binding and decoding the mRNA, which comes from nucleus to cytoplasm, and large subunits function is responsible for linking amino acid together and form proteins. Types of ribosome: one is 70S, other is 8Ts. 7Ts is found in prokaryotes and 8Ts is found in eukaryotes. 7Ts composed of 50S and 30S subunits. 50S is largest subunit and 30S is smaller subunit. And eukaryotes composed of, that is 8Ts composed of 60S subunit and 40S subunit. This is smaller unit and this one is larger unit. It is the image of two kinds of ribosome. 70S which is found in prokaryotes and 80S in eukaryotes. This one is smaller unit, this to this, and this one is larger unit, 50S. Okay, here 30S plus 50S. This is 70S and 80S. This is larger. This one is smaller unit, 80S, 60S plus 40S. Okay. Now come to vesicles. Many vesicles are found having different functions in the cell cytoplasm. Vesicles are small spherical membrane-bound sacs found in eukaryotic cells that function primarily in storage, transport, digestion of cellular materials. Okay, this is meant for digestion of cellular waste. Different kinds of vesicles are present. First one is transport vesicles. Move molecules like proteins and lipids within the cell, often from the endoplasmic reticulum to Golgi apparatus. Second kind of vesicles are secretory vesicles, contain materials like hormones, neurotransmitters, destined to be excreted from cell via exocytosis. Another process is also there, endocytosis. Example of secretory vesicle one is synaptic vesicles, which is present in the synapse. Lysosomes. It is otherwise called as suicidal bag, specialized vesicle containing digestive enzymes that break down waste, foreign invaders, and old organelles. Common in animal cells. In plant cells, lysosome is absent. Vacuole performs the function of lysosome. Peroxisome. Peroxisome break down the toxic substances and fatty acids using oxygen. Okay. Now come to vacuole, which is present in the plant and occupy larger space, similar to the vesicles but generally larger, functioning heavily in water regulation, especially in plants and storage. Okay, this is all about vesicles. Here one image. Vesicles are come from endoplasmic reticulum or Golgi apparatus. You can see this transport vesicles. Okay. Lysosome. This is the 2D image. Enzymes or proteins. This is the outer membrane. It is single membrane structure. This is the 3D image. Okay. This is all about cell organelles. In my next video, I will discuss about remaining cell organelles. Thank you.